to everybody for coming, and thanks to the previous speakers for helping work out the kinks and how to get the slides up here. So I'm the fortunate one who gets this done on time, but hopefully I won't mess it up by speaking too long. Today I'm going to talk to you about work from my lab where we're broadly interested in studying evolutionary transitions in behavior. Today I'm going to talk about one of my favorite behaviors, and that is brood parasitism in birds. Before I start, I'll just say that all of this work that I'm going to present has been done by a former postdoc in my lab and that of Mark Halber, and the three of us have been working on this together for a long while, and I've depicted there as Matt Lauder, who's now a postdoc at the University of Tokyo. So as you've probably been seeing already in this meeting and the previous talks, there's a lot of interest these days in the post-genomic era or whatever, and taking advantage again of convergent evolution to study the mechanisms of adaptation. And so this is a cool series of papers recently published in Philosophical Transactions. But a lot of results are starting to come out and highlighting sort of different patterns we see when we test for convergent evolution. So in some of our charismatic friends, we've seen evidence of molecular convergence at the level of the gene. In another study, using similar methods to what was just discussed, we've seen evidence not so much of convergence at the protein coding gene level, but at these regulatory sites. And we've also seen evidence of convergence at higher levels, sort of at the pathway level, where not necessarily the same genes are being repeatedly targeted, but the same pathways, they're different parts of the same pathways. And so I'm going to talk to you a little bit today about our attempts to look at convergence in brood parasitism, and I think brood parasitism is a particularly interesting set of traits in which to look at this. First of all, because this behavior has evolved 7 times independently in birds. And so obligate brood parasite is a bird that doesn't build its own nest or raise its own young, but lays its eggs in the nests of other birds. We have 7 independent origins, as I've said, and it's a nice context because these origins sort of span different time scales of evolution. So we have 3 origins within just the cuckoos and 2 just within the songbirds, but we also have these more divergent lineages that have independently evolved this behavior in birds. Additionally, brood parasitism is kind of really a suite of multiple traits that go together, incorporating multiple challenges that these birds have to face. For example, they have to be able to find host nests. They have to be able to avoid misimprinting on their hosts when they're young birds. In all of these cases, we have the loss of maternal behaviors. And in many of these cases, we also see accelerated growth of parasitic chicks relative to host chicks. Another interesting factor that sort of might factor into the likelihood of seeing convergence is the role of pleiotropy. And so when you're talking about behavior, we can think about hormones and their broad effects. And so you might think, okay, well, these birds are losing parental care. Maybe they're going to, their parental care related genes are going to degenerate. But most of these genes are highly pleiotropic, so maybe making that less likely. So today I'm going to try to squeeze in 2 little stories that we're working on in these birds. First, taking a comparative genomics approach, and secondly, an experimental study targeting one of those traits that I mentioned before, this issue of social imprint. So to begin to get at this, we sequenced a few new genomes to represent 2 origins of brood parasitism and 1 non-parasitic close relative, and we added that to a set of already published bird genomes. Bird genome, people are going crazy sequencing genomes at an alarming rate, so we don't have to do that much work, we just have to mostly sequence parasites. And so at this point we have 3 origins of parasitism represented. And then to start with, I'll talk about protein coding genes, where we were able to align about 8,000 orthologs across species. Me and a colleague at ECU, Michael Brewer, have been working on this pipeline because we're sort of neurotic about protein coding sequence alignments, and we think we're getting sort of okay at it, but I'll show you what's hard. And this is the punchline so far of protein coding genes. We really see no evidence of enhanced convergence over a null expectation. So basically here we're taking our three origins of root parasitism and saying, how often do we see convergent, positive selection? And we take three control lineages and say, do we have more or less? And there's really no difference between our control lineages and our target parasitic lineages. We do have some interesting storylines coming out, despite a lack of enhanced convergence. For example, we do see interesting signatures within, for example, individual lineages. And so in the prolactin receptor, we actually see, well, what we actually saw, we saw positive selection on the prolactin receptor in the Vidra clade, the indigo bird clade. But when we looked at that positive, that signature of positive selection, actually we found there's an error in the gene prediction. And so when we looked at RNA-seq data, what we actually find is that it's not accelerated evolution, but it's actually like a 5 amino acid deletion. So this. So rather than positive selection, actually we have an interesting deletion in the intracellular portion of the prolactin receptor. And so that's something that we intend to look at more closely going forward. We also have evidence of positive selection in the same lineage in thyrotropin releasing hormone TRH. Actually, in both cowbirds and indigo birds, we see kind of accelerated evolution, but statistically, it's restricted to the indigo bird. So these are two genes that we are continuing to do work on. So what about gene regulation? As in the previous talk, we've also taken the approach of trying to identify these conserved non-exonic sequences to identify putative regulatory regions. And the basic idea here, this is from that study on rat-type birds, we're looking for accelerated evolution in these otherwise conserved elements as a signature of relaxed purifying selection. And then again, we see the same thing that we saw in the protein coding genes. This is really, we see the same, basically, abundance of this loss of constraint in both our parasitic and control lineages. So again, we don't see any evidence of excess convergence in parasites relative to controls in these putative regulatory sequences. The last thing we want to look at is take these conserved non-exonic elements and look at them at sort of at the pathway level, the functional level. And here is actually where we begin to see maybe something interesting. We lifted these conserved elements over to mouse so we would have better annotation information and information on knockouts and things. And so at the level of gene ontology, we actually see much more convergence in our parasitic, our accelerated elements in parasites than we do in their controls. And the difference is less stark for these mouse phenotypes, but also there's a slightly higher degree of convergence in our parasitic lineages and also So far, we really see little evidence of parallelism at the molecular level, so protein coding genes or in regulatory sequences. But we do see this higher level of maybe pathway level convergence in the origins of parasitism. And so with that, I wanted to delve into one of these traits, as I said, in particular, this idea of the challenge that parasites face in avoiding imprinting on their hosts. And so as many of you may know, imprinting is an important part of bird life, particularly in songbirds. These birds learn to sing by copying their parents, basically, and so early in life, the song that they're exposed to determines the way that they communicate as adults. Our model system for this is zebrafinch. It's near and dear to my heart. And we know a great deal about how song learning works in zebrafinches. It's very stereotyped. It's a process that begins with auditory learning somewhere around day 20, and it concludes around day 90, where they have a fixed song that they'll sing for the rest of their lives. There is some plasticity in this, but relative to many other species, this this is our timeline for song learning in these birds. And so this, we know, because of all the work on zebra finches, we know a great deal actually about the neural substrates that underpin this song learning behavior. But it raises a major conundrum, I would say, for brood parasites, and that is how do brood parasites accommodate this problem? They're being raised by different species. Why doesn't the baby cowbird imprint on his foster parents and sing their own song, basically? And so it turns out that in around the early 2000s, 2001, Mark Halber proposed this idea that he termed the password hypothesis for species recognition. And the basic idea here is that baby cowbird has to be exposed to an innate conspecific cue, and hearing that cue triggers learning of the rest of cowbird's song, even potentially phenotypic plumage color traits. And so since this time, we haven't really been able to get a handle of, does this really happen? And so that's what we've been working on for the last few years. And so the way Mark proposed that a chatter call given by female cowbirds could serve as the password for species recognition in cowbirds. And so what we wanted to ask is, does hearing this password, this chatter call, trigger neuroplasticity in the brain and learning of song? And so we had to learn how to raise baby birds. I'd never done that before, and it was fun. It took a lot of us and a lot of work. And we did some experiments with those baby birds that I think are work quite fun. So yeah, I'll describe quickly an experiment in males and an experiment in females. In males, basically, we had two groups. And basically what we were trying to do is get them to learn canary song. And so some males were exposed to canary song that was paired with the chatter, and they think might be the password. And other birds were also heard canary song, but then they heard dove with that. So they were never exposed to the chatter. And then the day that we euthanized these birds, we played them canary song and looked at what happened. We measured gene expression in the auditory forebrain, which is known to be involved in song. First of all, the crazy thing is that the experiment actually worked, and the birds that were, yeah, that's great. The birds that were exposed to the chatter actually sang more canary-like syllables than the birds that never heard the password. Secondly, we had really strong signals in the brain of differences in brain development. We had really strong gene ontology enrichment and a number of really cool genes with known roles in neural development, suggesting that this chatter call really does induce neuroplasticity. In females, we did a different and more complicated experiment where each female was played two different canary songs, so structurally different canary songs. And then one of those songs was associated with the chatter, so they were played back to back. And the second canary song was associated with the dove coo, so salient, but not the cowbird song. And then on the day that we sacrificed these birds, they either heard that canary song that had been paired with the chatter, or they heard the other canary song that they heard that was paired with the dove. And here we saw really interesting signatures, and the way we're thinking about this is that we saw a signature of habituation. And so basically birds that had been that were played the chatter paired canary song at the end showed lower levels of neural activation in the auditory forebrain. So this is a plot of PGR1 called Zinc by bird people, and we get higher expression of this gene in the control birds than the other birds. And the same is true for 4 other of these immediate early genes, these genes involved in neural activation and memory specifically. And so we have this really strong signature of habituation consistent with learning of this, enhanced learning of this chatter-paired canary song. So basically, we really find Oh, I didn't hear the comment. Sorry. Thank you.